In this video, we're going to take a look at what happens as antigens and antibodies react together and how that can be used, or how that reaction then is used to help protect the body. So antibodies protect, the kind of reactions that they can do, protect by defending the body against bacterial infection, by neutralizing toxins that are found in the body, and by inactivating viruses that haven't yet entered the cell. And the reaction, or the antibodies themselves, are found in different locations. IgA is found in tears, in secretions that come, that are produced in the nasal passages, in saliva, and in other body fluids, helping to protect the orifices, the openings into the body. In order for bacteria to infect the body, then, the bacteria has to be able to bind to surfaces, to the cells, to the surfaces of the cells of the body. And so the way IgA antibodies protect is that they coat the bacteria and they cover up the proteins that would have been used to bind that bacterium to the body cells. And in this way, that prevents the bacteria from being able to bind and infect. IgE, these are antibodies that are produced by the plasma cells that are found in lymph nodes, in the gut-associated lymphoid tissues, and in other lymphoid tissues that are found in the body. And they bind to mast cells, and as that bound IgE encounters antigens, then the mast cells release histamines. And then histamines initiate and accelerate the processes of inflammation. And then as part of all that inflammation, IgG and complement proteins are delivered into the infection site. So as we bring in more blood, it brings in IgG and all the complement proteins. And that then triggers all the effects that we see associated with IgG and complement. So as antibody binds to antigens, then we see that the microbes that the antigens are found on are all inactivated in these ways. Through agglutination, which is when microbes are all kind of glommed on to microbes with antibodies into big large clumps. IgM is capable of producing very significant agglutination reactions. Remember, IgM is found in body fluids as that a pentamer, so there's 5 of them, and so we've got the capacity. One IgM complex can bind up to 10 antigens. IgG produces weaker but still significant agglutination reactions. Opsonization, that's when the antigen is coated and then phagocytes are triggered to come and consume it. Activation of complement, IgG and IgM both cause a strong complement reaction and initiate the cascade of complement reactions with all of the effects that we've seen associated with complement. Lysis of the cell, so IgM can actually directly break open cell membranes. And then with neutralization, when the antibody binds to the antigen, it can block the effect of a toxin and or prevent binding by the antigen to any body cells. So IgG is the main neutralizer of bacterial toxins that are produced, and IgM, IgG, and IgA all can neutralize virus particles. So what are some ways then that antibodies can be produced in the lab or for therapeutic use? We do this by producing in the lab a cell that's called a hybridoma. It's a hybrid of a B cell that's been sensitized to a particular antigen, and a cell called a myeloma. These are cancerous immune system cells that are kept in cultures in labs. And so if you can hybridize this B cell with the myeloma, then you can produce an antibody-producing cell that will continuously divide, producing more antibody-producing cells. And so those cells, the offspring cells, will produce antibodies that can be used both for research and for therapies. They're referred to as monoclonal antibodies, the antibodies they make, because it's a big clone of the same, of a single antibody producer. That's why they're called monoclonal antibodies. And they can be used in diagnostic tests because as the antibodies bind to antigen, we can find evidence of them. And so they're used as pregnancy tests, and they're effective as early as 10 days post fertilization of the egg. And they can be used to diagnose hepatitis, flu, herpes viruses, and chlamydia. So because they are so very sensitive, and it doesn't take a lot of antigen to be able to find that antigen-antibody reaction. And they can also be used to target specific cancers and treat those cancers. So they've been used for non-Hodgkin's lymphoma and for breast cancer. So if you can attach a drug or some radioactive material to an antibody that is very specific to breast cancer cells or to these non-Hodgkin lymphoma cells, then you can introduce that into the body. The antibody binds to the cancerous cells and delivers the drug, or very in a targeted way delivers radioactivity to that cell and delivering the treatment. So very exciting potentials for treating diseases come from these monoclonal antibodies as well. Okay, so that's everything. It was a shorty, so I hope you enjoyed that.